I was just reading the latest clinical data on metabolic cofactor decline in aging cells, and it changes how we should evaluate cellular repair decay. Our cells face constant daily damage. We often assume that cellular aging is an inevitable passive wear and tear process. However, the biological reality suggests it is closely linked to a drop in metabolic repair capacity. Here is the mechanistic question. How does a cell lose its ability to fix daily damage? By looking at metabolic cofactors, we can see how the cell's repair systems decline. Every cell in the human body requires substantial metabolic energy to repair its DNA and maintain structural integrity. Without sufficient metabolic support, these repair systems gradually slow down and eventually fail. This metabolic slowdown is not just a general decline. It represents a specific bottleneck where the resources needed for maintenance are no longer sufficient to keep up with incoming cellular damage. Understanding this process helps us answer a fundamental question. How does aging reduce cellular repairability? The answer lies in the availability of key metabolic cofactors that drive these repair systems. In the aging cell, the loss of repair capacity clearly manifests as accumulated damage. Over time, unrepaired DNA lesions and damaged proteins accumulate, leading to progressive cellular stress and functional decline. This cellular decay is not merely an aesthetic concern. It affects vital organs, particularly the human brain, where highly active cells depend on constant repair to maintain their complex signaling networks. When repair systems cannot keep up, the cell enters a state of persistent stress. This stress can disrupt synaptic communication and impair the overall efficiency of neural networks in the brain. Follow the evidence boundary. We must always distinguish between the symptoms of aging and the underlying metabolic mechanisms. The primary issue is a decline in the cell's internal repair efficiency. As cellular repair efficiency drops, cells become increasingly vulnerable to external stressors. Normal daily physical activities can create a level of cellular damage that the cell can no longer adequately resolve. This vulnerability creates a progressive cycle of decline. Unrepaired damage impairs metabolic pathways, which further reduces the chemical energy available for future repair, accelerating the overall aging process of the cell. To understand this metabolic decline, we must define the role of nicotinamide adenine dinucleotide, or NAD+. This cofactor is essential for the function of enzymes that repair DNA and regulate cell survival. NAD+ acts as a critical link between energy production and cellular maintenance. It serves as a necessary cofactor for sirtuins, which are proteins that regulate cellular health and stress responses. Additionally, NAD+ is consumed by PARP enzymes, which detect and repair DNA damage. When DNA damage occurs, these enzymes consume NAD+ to activate the necessary molecular machinery for genomic repair. In a young, healthy cell, NAD+ levels are high, allowing both energy production and DNA repair to proceed efficiently. The cell easily maintains a dynamic balance between these two competing demands. However, as cells age, NAD+ levels gradually decline. This metabolic decline is driven by both decreased production of the cofactor and increased consumption by chronic, age-related cellular stress pathways. When NAD+ levels drop, the activity of both sirtuins and PARP enzymes is constrained. The cell must choose how to allocate its remaining metabolic resources, often prioritizing survival over repair. This resource allocation shift leads to a gradual accumulation of unrepaired cellular damage. The cell's ability to maintain its genome and protect its active mitochondria is significantly compromised under these conditions. So, the metabolic clock of aging is directly linked to the decline of this vital cofactor. Let us explore the molecular steps that connect NAD+ depletion to lost cellular resilience. Now, we must trace the core mechanism linking NAD+ depletion to impaired cellular repair. The process begins when chronic low-grade inflammation activates enzymes that consume massive amounts of vital NAD+. These consuming enzymes, such as CD38, deplete the cellular pool of NAD+. This metabolic depletion leaves less of the cofactor available for the critical repair enzymes that maintain normal genomic stability. With less NAD+ available, PARP enzymes cannot respond effectively to DNA double-strand breaks. These breaks remain unrepaired, leading to cellular mutations and activating pathways that can eventually trigger cellular senescence. Senescent cells stop dividing but secrete inflammatory molecules that damage neighboring cells. This localized inflammation further increases NAD+ consumption, creating a self-reinforcing loop of metabolic decline and cellular damage. At the same time, declining NAD+ levels impair sirtuin activity in the mitochondria. Sirtuins, particularly mitochondrial sirtuins, are crucial for maintaining mitochondrial efficiency and controlling toxic reactive oxygen species. When mitochondrial sirtuin activity drops, mitochondrial function declines and the production of reactive oxygen species increases. This increase in oxidative stress creates even more DNA damage for the cell to repair. This combination of increased DNA damage and decreased repair capacity leads to a rapid loss of cellular resilience. The metabolic cofactor decline directly constrains the cell's ability to recover from stress. Neurons are particularly sensitive to this decline. Because they are non-dividing cells that must last a lifetime, they rely heavily on constant DNA repair and mitochondrial maintenance to function and survive. In the brain, impaired mitochondrial function reduces the energy available for synaptic transmission. This metabolic limitation can affect overall cognitive performance and contribute to the gradual loss of synaptic density. Separate the mechanism from the outcome. We are observing a metabolic constraint on cellular repair, not a programmed destruction. The cell is simply running out of the cofactors needed for maintenance. This distinction is crucial for developing logical models of cellular aging. By identifying the cofactor bottleneck, we can easily understand why repair capacity drops so dramatically in these older cells. As the bottleneck tightens, the cell's internal environment becomes increasingly disorganized. The loss of mitochondrial efficiency reduces ATP production, which is the primary energy currency for all active cellular processes. Without adequate ATP, active transport systems within the cell slow down. This slowdown affects the distribution of nutrients and the removal of cellular waste, further compounding the cellular stress response. The cell enters a state of metabolic crisis, where the priority is short-term survival rather than long-term maintenance. This shift in priority is the key driver of cellular resilience decay. Over time, this persistent state of low energy and accumulated damage leads to the loss of cell function. The metabolic clock of aging is therefore a measure of repair capacity decline. The important limitation is that this metabolic model is primarily conceptual and based on laboratory observations. We cannot assume that these cellular pathways operate identically in all human tissue types. Furthermore, a counterpoint exists. Some cells maintain high repair capacity despite lower NAD+ levels. These cells may utilize alternative salvage pathways to recycle cofactors, showing that depletion is not universal. This variation suggests that genetic differences and local tissue environments can influence how cells respond to metabolic decline. Depletion is a trend, not an absolute rule for every single biological cell type. We must also consider that temporary metabolic stress can sometimes trigger adaptive responses. In some cases, mild cellular energy depletion can activate backup repair systems, temporarily increasing overall cellular resilience over time. A validating case is observed in yeast and rodent models, where supporting these metabolic pathways can preserve cellular repair. However, these animal studies do not guarantee similar outcomes in humans. We must remain cautious when translating these laboratory findings into clinical expectations. The complexity of human biology means that simple interventions rarely produce the straightforward results seen in controlled clinical research. Therefore, we must evaluate metabolic cellular repair within its clear biological limits. Understanding these boundaries prevents us from overgeneralizing simple cellular mechanisms to complex whole organism outcomes like overall human health span extension. What matters clinically is how this metabolic decline explains the reduction in cellular repair. By connecting NAD+ depletion to DNA repair enzymes, we find a direct link between metabolism and aging. The metabolic cofactor decline acts as a constraint, limiting the rate at which repair enzymes can function. The cell does not lose the enzymes themselves, but rather the fuel they require. This explains why cells in older individuals accumulate damage even when the genetic instructions for repair remain intact. The machinery is present, but the metabolic cofactor levels are insufficient. By focusing on this cofactor bottleneck, we resolve the mystery of cellular repair decay. The aging cell's loss of resilience is primarily a direct consequence of metabolic resource and cofactor depletion. As NAD+ levels fall, the balance shifts from active repair to survival-oriented processes. This shift is what ultimately reduces the cell's capacity to maintain its biological structure and normal function. This metabolic constraint is particularly evident in the brain, where the high energy demand leaves little margin for error. Small drops in cofactor availability can have noticeable consequences for neural health. Understanding this link allows us to view cellular aging through a more precise lens. It is not a random accumulation of errors, but a predictable response to metabolic resource limits. This perspective reframes our approach to healthy aging. Instead of trying to prevent all daily damage, we can focus on how cells manage and distribute their limited metabolic resources. Ultimately, the decline in cellular repair is a problem of resource allocation. Resolving this metabolic bottleneck remains the central challenge in preserving cellular resilience as we grow older in age. To apply these concepts, we must focus on lifestyle factors that naturally support metabolic pathways. Low-risk health behaviors can help maintain the cell's internal environment without introducing unproven interventions. Optimize the protocol. Regular physical activity and structured sleep patterns have been associated with improved metabolic efficiency. These basic habits support the cell's natural salvage pathways over time. Additionally, nutritional patterns that avoid chronic calorie excess can help reduce systemic inflammation. Lower inflammation levels reduce the activation of enzymes that deplete vital metabolic cofactors. Circadian alignment is another important consideration. Aligning daily light exposure with natural cycles supports the rhythmic production of cofactors, helping cells maintain their repair capacity over time. These general health behaviors do not offer a simple way to reverse aging. Instead, they provide a stable framework that allows cells to distribute energy more efficiently for repair. We must avoid the temptation to seek quick solutions through unproven supplements. The science of metabolic support is still developing, and individual responses can vary significantly. When considering any meaningful change to your routine, it is important to consult a qualified health professional. This ensures that your approach is safe and tailored to your specific needs. By focusing on verified low-risk habits, you can support your cell's natural repair mechanisms. Preserving metabolic efficiency is a gradual process that requires consistent daily effort. We must continue to monitor the clinical evidence as it emerges. Optimize the protocol, protect your brain, and I'll see you in the next data.